Welcome everyone to another beautiful LatinX speaker series. My name is Johnny Pena. I am part of the LatinX Shades at Broad. Today is September 20th, and we have another very wonderful speaker series for you today. Just a little bit about the LatinX group here at the Broad. LatinX at Broad has the mission of building community among Latinx Broadies and allies. The LatinX community is an affinity group at the Broad with the mission to bring people together to empower them to reach their full potential. At this very moment, we need a lot of people in the LatinX steering committee, because if you can only imagine so many things that are going on, especially with the Latinx speaker series, there's so many hands behind the scenes, but we welcome everyone and part of the Latinx community to reach out to us and actually just get informed, see how you can help, and we'll get you acquainted with everyone and situated in that aspect. Oops, sorry. Our Latinx speaker series, what is it? Our Latinx speaker series, we highlight Latinx and diverse researchers within and outside the Broad, and we foster a lot of collaboration. Following this mission, we created a Latinx speaker series with a purpose of the hope to expand and to expose you all to the different types of research that is going on within the Latinx community. Some that may be oversighted and some that may just be up and coming. We hope at the end of this program, people will take from this with a better understanding of the research environment in the Latinx community. With this out the way, we do have, with this opportunity, we will go ahead and move on to a speaker that I am so happy to introduce to you all shortly. The speaker that we have today, she goes by the name of Janitza Montalvo-Ortiz, PhD, and what she will be going over today is decoding the epigenomic landscape of trauma across lifespan. A little bit about Dr. Montalvo-Ortiz. She's a she is a neuroscientist and assistant professor at Yale School of Medicine. She obtained her BA in biology at the University of Puerto Rico, Rio, sorry if I butcher this, Rio Piedras Campus, and her PhD in neuroscience at the Northwestern University at Chicago. Her NIDA and VA-funded lab utilizes computational approaches to the study of the genomics and epigenomics of the neuropsychiatric disorders with a focus on trauma and substance use abuse. She is the recipient of multiple awards, just a little round of applause on that portion, including the VA Career Development Award, the NARSAD, which I'm pretty sure you'll go into more as you speak, Young Investigator Award, the Faculty for Excellence and Diversity Initiative Fellowship, and the Lawrence Family Young Investigator Award. She also serves as the co-founder and co-leader of the Latin American Genomics Consortium and the Psychiatry Genomics Consortium Substance Use Disorders Epigenetics Workshop. Without further ado, Janitza, I'm going to pass it off to you and let you share your story.
Okay. Thank you so much, Johnny, for such a nice introduction. And for me, this is an honor to presenting the Latinx and Spanish Heritage Month. And I think it's such a great idea putting this seminar series together to, allow us to gain visibility in the field. Thank you so much for having me. As Johnny said, I'm an assistant professor in the Department of Psychiatry, Division of Human Genetics at Yale School of Medicine. I'm also at the Connecticut VA, and I'll be presenting our work titled Decoding the Epigenomic Landscape of Trauma Across the Lifespan, where I'll be presenting different pieces of published and unpublished work that I hope you find interesting. So just to go briefly over my career path in case you have any questions or you want any advice on how to get to a tenure track faculty in the academia. coming from Puerto Rico or underrepresented minority group. So my trajectory looks pretty linear, but yeah, has been events where it's kind of uncertain where to go, but luckily it all started from Calle, Puerto Rico. It's a small town in Puerto Rico in the mountain area, and I show a picture because it's so pretty. And I did my bachelor's degree after graduating from high school from the public system in the University of Puerto Rico in San Juan, and then I was admitted to do a PhD in neuroscience in Northwestern University in Chicago, Illinois, and started my postdoc working under Joel Galenter and John Kaufman at Yale University. And luckily, I was promoted to assistant professor and started my own lab since 2019. So just to go over the outline of my talk, I'm going to start a little bit about the background on defining trauma exposure and how it relates to the development of disorders, going over why there is an urgent need to identify biomarkers of trauma-related disorders, and present epigenetics as one of the biological mechanisms that may underlie the long-standing effects of trauma exposure and later development of disorders, focusing mainly on DNA methylation. And we're going to ask then a series of questions. So the talk will be question-driven, that I hope makes sense and help the talk to be fluent. So the first question I'm going to ask is very simple, very straightforward, and is that whether epigenetic changes are associated with trauma exposure or post-traumatic stress disorder. And we're going to go over data from children cohorts and adult cohort. After that, we're going to ask the question, are trauma or PTSD-associated epigenetic changes long-lasting? Since these are very dynamic biological mechanisms, there's a question whether or not these are long-lasting. That could be underlying development of disorders later in life. And also followed by the question, can epigenetic changes be reversed? And we're going to show very interesting data on a poorly understood mechanism, but very interesting, called DNA hydroxymethylation. We're going to show parallel findings with DNA methylation in human postmortem brain. We're going to finish with the question whether epigenetic biomarkers that we identify in multi-omic data sets could be used to inform treatment for trauma-related disorders, which is the endpoint that we always try to get at when doing this kind of research. So we know that 50 to 60% of the population experience trauma over the course of their lifetime. However, not everyone exposed to trauma develop trauma-related disorders. Actually, it's very few portion of the people exposed to trauma that develop health disorders. Trauma exposure is known to lead to later development of post-traumatic disorder and also increased risk of other physical and mental health disorders. And I'm going to explain why there's an urgent need to identify biomarkers, and is that because there are challenges in diagnosis and treatment due to stigma from people suffering from trauma. There are limitations of identifying those at risk, and a wide scarcity of tools to predict treatment outcome. So we're going to try to identify biomarkers at every single stage: prevention, prognosis, and treatment. And in order to do that, we have to understand that trauma-related disorders are multifactorial, multisystemic, and it can impact multiple biological domains such as genes, proteins, cells, tissues, and organisms, sort of like the RDUG paradigm. And this biomarker discovery must occur beyond a single domain study design. So I know current research is focused on looking at the genome, transcriptome, epigenome, proteome, but research needs to be moved towards multidimensional and integrated omics studies, ideally within subject design. So hopefully by identifying multi-omics biomarkers via the integration of information gathered from multiple domains and using these in unbiased approaches and willpower sample, we'll be able to improve prevention, diagnosis, and treatment outcomes in people that suffer from trauma-related disorders. And in terms of this talk today, I'm going to focus on epigenetics. And the reason why we are particularly interested in epigenetics is because these mechanisms may underlie the interplay between genetics and environmental factors, potentially having trauma exposure playing an important role in how it may interact with our biological factors to increase the propensity to suffer from later on from disorders. And within the epigenetics, we're going to focus on DNA methylation. DNA methylation is defined as the attachment of a methyl group to the DNA. And presence of methyl groups has been related to transcription repression, where absence of methyl groups transcription to transcription activation. So presence or absence of methyl groups can dictate whether transcription processes are going to occur or not, playing a major role in gene regulation. So in regards to our first question, whether epigenetics changes are associated with trauma, we're going to first talk about our data from children cohorts in early development. So trauma exposure in early life is important to study, given that since based on data from the Global Status Report on Preventing Violence Against Children from 2020, around 1 billion children worldwide suffer from some form of violence each year. And there are four main types of how trauma exposure can occur during childhood. Could be in the form of sexual abuse, physical abuse, psychological abuse, and neglect. And childhood trauma has been strongly associated with later development of negative mental and physical health outcomes, including obesity, personality disorders, depression, aggression, and substance use disorders. And recent work has shown that epigenetics may mediate the long-lasting effects of childhood trauma. And I'm going to show here our data from different epigenetics studies that we've done looking at trauma in children cohorts. The first study titled Adverse Childhood Experiences, Epigenetic Measures and Obesity in Youth. The second one is titled Methylation in OTX2 and Related Genes, Maltreatment and Depression in Children. And the third one, DNA Methylation and Trauma Interactions, Identified Signatures of Aggressive Behavior in Maltreated Children. The first two studies are already published work, and the third one it's under review. So in terms of the children cohorts, the data that I'm going to show, these are basically mostly based on the Vermont cohort. This is a cohort of 168 children, 50% of them female, 95% of them European Americans. These are all from the from socioeconomic background, from similar neighborhoods. The trauma exposure that we're gonna look at is intrafamiliar trauma, and we examine different variables, including body mass index, child behavioral checklist, and mood and feeling questionnaire. We collected saliva samples for DNA specimens, and a subgroup of this cohort, we also had MRI data available. And I'm here showing the pictures of how this data was collected. So it was a very nice summer camp setting in Vermont, where we had here the different cabins where psychological assessments were performed with the kids. And this was a summer camp as any other summer camp for kids, in that they were, doing fun summer camp activities such as paddleboarding, archery, and in the university, then of Vermont, then they also participated in MRI and other imaging assessments as well. So for that DNA from saliva was used for DNA assessment, DNA methylation assessment. And we did this using the Illumina 450K bedsheet array, where we conducted an epigenome-wide association study. So just going briefly over how these data looks like for a given sample, we have the following data. So we have a cartoon of each of the chromosomes, 1, 2, all down to 22. And this is showing how the data could look like in terms of on a single loci. We could have a value ranging from 0 to 1, 0 close to be unmethylated and 1 to be methylated. And we have a wide range of methylation values, and these values are actually in the form of beta values defined as methylated probes divided by the total probes and the offset error value. And the model that we used in the context of the 2 studies that I'm going to show that looked at obesity and aggression is that we used the trait of interest as our outcome variable as a function of DNA methylation and also the interaction term with intrafamilial trauma and the covariates. We also use a random variable of family ID just to account for the relatedness in our cohort, given the amount of children that are siblings or cousins. So this is the first study that I'm going to talk about, focus on obesity, where we were able to identify DNA methylation signatures that were significant with the methylation term itself, but also in the interaction term, interaction between methylation and trauma. And from these, we were able to replicate some of our signals in a Connecticut cohort that is a more diverse cohort in terms of ancestry, but also from low socioeconomic background. And it's important to be able to replicate findings from any discovery cohort, but also to try to replicate it to more diverse samples when coming from a homogeneous, mostly European-based cohort, and to ensure that these findings could be translatable across ancestries. So from these genes that were identified, we were able to replicate the PCK2 and HID1 and GALE and others that didn't map to genes. And from those, we did a sort of mediation analysis, a series of mediation analysis. So we identified that the association between interfamilial adversity on BMI was mostly driven by and moderated by methylation changes at the PCK2. And this gene is very interesting because the gene has been previously implicated in obesity risk in a prior published EWAS study. So observing the interaction effect with trauma and observing this effect in children cohort, it's very interesting because it gives insight on potential long-lasting effects. So we also look at childhood aggression, and this was, we started with the subcohort of 105, where the data was gathered from the biological mothers. And sort of giving an idea of a less severity of trauma exposure. And we found that by looking at aggression using the Child Behavioral Checklist, intrafamilial trauma was a significant predictor of aggressive behavior reflected as CBCL T scores. And we identify several hits associated with aggressive behavior. One of them, I'm going to point out one of them mapping to the HBCAL4. which was replicated in an independent Connecticut cohort, more diverse cohort. And we observed that childhood aggression was associated with decreased HPACL4 methylation. And in a subset of children, we look at brain functional connectivity and observe that brain functional connectivity was associated with HPCL4 methylation and aggression scores. We also look at on a bigger cohort, including kids where we have data from foster care system, which are kids that are that show a more severity of trauma history. We found that we found an interaction signal with mapping to FSD1 gene, and also several hits with the methylation term associated with aggressive behavior. And from this, we look at replication in the independent Connecticut cohort, and we found that these 4 genes, APOB, GRIA1, PSAEN1, and KSOX1 were replicated in the independent cohort, with APOB and GRIA1 surviving multiple testing, and KSOX1, sorry. And from these genes, I want to highlight 2 of them, which have been shown previously implicated either in aggressive behavior, as with the case of APOB, and also psychiatry disorders, as with the case of GRIA1. We also look at, we try to leveraging the data of children cohort that have trauma history. We look at a recent paper then from Netzler's group and Peña's group, where using an early life stress mouse model, they identified OTX2 downregulation to be associated with early life stress in mice. And we wanted to sort of replicate those findings in animal models to humans, and using the children cohorts. And in the 157 children, and using the depression data assessed with the mood and feelings questionnaire, and sort of using the same models as shown before, we found that OTX2 methylation was associated with depression, sorry for the typo, in children exposed to trauma. And this actually, the changes in the patterns of methylation at these genes was actually also related to connectivity using resting state functional MRI data within the medial prefrontal cortex. So basically, we show that increased methylation of OTX2 was associated with increased functional connectivity among different brain regions that are very well known to be implicated in depression. So we here show changes that occur in saliva in terms of epigenetics, how these correlates with changes at the functional level in the brain. And we also look at genes that are regulated by OTX2 gene, and we found also associations with WNT1 gene, which is known to play a big role in the establishment of midbrain dopaminergic neurons, and to be a major regulator of neuronal plasticity. So just to conclude the first part of the talk, we found and we've proven, also it's consistent with the literature so far, that alterations of DNA methylation are associated with trauma exposure in children. And we show new data on looking at intrafamiliar trauma, which is a more severe case of trauma history, and mapping changes that occur in the periphery in terms of methylation patterns to those changes, functional changes in the brain. So we observe interactions between intrafamiliar trauma and DNA methylation. This is also novel in the literature. And we found that these may underlie the development of health risk and maladaptive behavior. Specifically, the effects of intrafamiliar trauma on indices of obesity, We found that it's mediated by epigenetic changes in the PCK2 gene, a gene that has been previously implicated in obesity risk in a prior EWAS. And we also identified epigenetic signatures of aggressive behavior in children exposed to maltreatment. Some of these replicated in interaction with trauma exposure, including APOB and GRIA1, genes that has been known in the literature implicated with aggressive behavior and psychiatry traits and disorders. And as observed in a mouse model of early life stress, we also show that methylation in OTX2 and the regulated genes such as WNT1 is associated with depression and increased functional connectivity in key brain regions implicated in depression. So those are results shown in children cohorts. Now I want to show our studies done in adult cohorts. For this, we're going to focus on post-traumatic stress. So trauma exposure is known to lead to the development of post-traumatic stress disorder. And this PTSD is known to be characterized by intrusive symptoms, avoidance, negative cognition and mood, and alterations in arousal nerve activity. There is a lifetime prevalence of PTSD in around 8.7%. And while recent large-scale GWAS studies, genome-wide association studies have found genetic risk factors associated with PTSD, these still only explain a small portion of disease liability, implicating additional mechanisms involved. And recent work in humans has investigated a potential role of epigenetics in PTSD. And I'm here showing a table from a book chapter that I recently published, where I summarized all the work so far done in cross-sectional epigenomic studies of PTSD. And here I'm including this table just to show sort of the history so far on the EWAS of PTSD in humans, sort of like, and also the evolving work in terms of increasing sample sizes, but still pretty homogeneous in the type of tissue that is analyzed, and pretty limited also that most of the work has been mainly in 450K array, which compared to the newest array available, the EPIC array, it's still, that basically has double the size of probes that are examined for methylation changes across the genome. And I'm here summarizing the main findings, mainly across the studies, they've been pretty unsuccessful at identifying genome-wide significant hits. Some of the findings has been at the pathway level. We do have some small studies showing replication, but this needs to be assessed in bigger cohorts. And some of the latest research and later studies with bigger samples, mostly from the PGC PTSD Workgroup, Epigenetics Workgroup, has identified a HRR gene, but this is a gene also very well known in the smoking literature. So we need more data, bigger cohorts, or at least, cohorts that are carefully ascertained for PTSD that would allow us to increase that power to get to those significant hits. So this is our new study recently published in Translational Psychiatry, where we did an EWAS in the National Health and Resilient in Veteran Study cohort. We included in a single cohort over 1,000 subjects, all European American, male, US military personnel, which is limited by being pretty homogeneous in terms of ancestry, sex, and type of trauma, but we hope to be able to expand these findings to cohorts that reflect those of general population. So PTSD was assessed using the PTSD checklist specific, and we assessed both lifetime and current PTSD. DNA specimens were collected from saliva samples. This is the first study looking at saliva, and we used the latest version of the human methylation bed chip array, the EPIC array, that assessed 850,000 or so probes across the genome. And in terms of the model, pretty much the same model that we use for the children cohorts. So here I'm showing the results for the current PTSD EWAS. I'm showing here on the left panel, upper panel, the Manhattan plot. So for those that are not familiar with the Manhattan plot, we basically show the data for each of the probe or loci across all the chromosomes 1 to 22. What we have in the x-axis and in the y-axis, we have the p-value. And just to be mindful of the multiple testing correction, we use the most stringent threshold, the Bonferroni threshold, which is the line up here, and the dotted line shows more loose threshold as FDR, which is also pretty common to use in EWAS studies. And our top hits are those that are above this line. And here I'm showing those hits that we observe up here, mapping to this 6 genes. And we look at, since PTSD is a very multifactorial disorder, we look at the symptom level, how these different hits and signals were driven or potentially driven by specific symptoms in PTSD. And while we observe some potential specificity for some of our hits, like in the case of RAMP3 for re-experiencing of PTSD, some were pretty much across the board, like the CG155 and the one mapping to DYNC1H1. In terms of lifetime PTSD, we identified 3 different hits. And again, we're looking at the symptom level. We actually here see pretty much effect across the different symptoms of PTSD for all 3 signals. We also asked the question of whether or not these changes in methylation were driven by genotype variation. And for this, we conducted a methylation quantitative trait loci analysis. And we did a linear regression model just as the same way we conducted our EWAS, and including the same covariates. And we analyzed over 12,000 SNPs in a window of 50 kb. And we look at those 9 genome-wide significant CPG sites. And here I'm showing the sort of the different SNPs in the different mapping to the different CPG sites and the p-value, and found a total of 30 MEQTLs identified, and mapping to the CG155 nearby the ETS1 gene, a gene that has been involved in immune regulation. So this shows that some of our differential methylation signals may be driven by our genetic background, but some others may be just on its own. So in terms of replication, this is very important. Every time we conduct a genome-wide analysis, from our 9 hits of both current and lifetime PTSD, we look at replication in an independent cohort of 608 male veterans from the BACS biobank cohort, and we identified that methylation levels of SEMP7 was replicated and were found to be decreased in the same direction effect as in the discovery cohort with a p-value of 0.02. And we also look, I'm also a neuroscientist by background, by training, sorry. So I'm always asking, well, are these changes that we're observing in the periphery are reflecting or telling us anything that's going on in the brain as well? Because we have to keep in mind that these are brain disorders as well. So we look at human postmortem brain tissue, and even though we didn't have methylation data available, we look at RNA sequencing data to sort of seeing whether they were differentially expressed. So we look at SENP, and we found that we observe a significant decrease of this gene in PTSD brains in a total of 35 brains from the medial orbit or from the cortex, which is data that I'm showing on the right. And this gene is actually very interesting. At first, you see, okay, it's involved in transcription regulation. Itself, it's interesting, but still, it's quite uncertain how it's connected to PTSD. But looking at the GWAS catalog, we found that there were genetic variants associated of this gene associated with risk-taking behavior and alcohol consumption. And given the somewhat correlation with PTSD of these traits, it's quite interesting seeing a potential role of this gene in PTSD. So to summarize the second part of my talk, we show that alterations of DNA methylation are associated with PTSD in adults. So this is consistent with what we found and what I showed in the table, summarizing all the cross-sectional studies, but also showing new data with saliva samples as the first study assessing saliva. And saliva is interesting because it's a very easy, accessible tissue, non-invasive, and research shown that it's actually more highly correlated to changes in the brain than those in the blood. So it's a very good candidate for identifying biomarkers. We identified differentially methylated sites for both current and lifetime PTSD. That's also novel in the literature. And some of the observed differential methylation are influenced by genotype variation as observed with the 30 MEQTLs identified. The findings from SCNP7 were replicated in an independent veteran cohort, and we observed that expression changes of this gene were decreased in the human postmortem orbitofrontal cortex of PTSD cases. This gene is promising. It's involved in transcription regulation, but also it has been associated with risk-taking behavior and alcohol consumption. So another question that I wanted to ask is whether these changes are long-lasting. And here I'm just showing what's in the literature so far in looking at longitudinal changes of DNA methylation associated with PTSD. So far, there has been 4 different studies, and the sample sizes range is pretty much between 200 and 300. And I'm mentioning this because in the cohort that we showed the data of the EWAS of PTSD in saliva in veterans, we were able to follow up this cohort for different waves. So the baseline was the data that I showed in 2011. And this cohort has been assessed in the 2 years after that, 4 years, 7 years, and also in 2021 last year, where was the completion of a 10-year period. So this was possible because of a career development award from the VA that I am recipient of. And we look at how the different trajectories of PTSD symptoms changes over time. And we identified 2 different groups, one that showed no lung symptoms and one elevated symptoms. And basically, we observed different trajectories and whether there are no changes across time or decrease as is expected in the literature. And hopefully I'll be able to show data on this soon. We were just finalizing the assessment of all 300 subjects from the 1,000 ones, but this is after 10-year follow-up. So it was a success on its own. And it's gonna be comparable sizes to the literature in terms of longitudinal studies. And different from the literature, this study is actually one of the longest longitudinal study in a 10-year follow-up. So yeah, I'm looking forward to see what the data looks like in terms of methylation, and also we'll be able to integrate with genetic data and a transcriptomic data. So that will be upcoming. And then the next question that I wanted to ask is whether these changes that we're observing at the epigenetic level could be reversed, sort of like guiding us to potential, early interventions efficacy, or treatment efficacy for trauma-related, for people that suffer from trauma-related disorders. So we're looking at the DNA methylation and how this process could be reversed. There was some work done by biochemists into elucidating how DNA methylation wasn't actually a stable, non-dynamic modification as it was originally thought. It was actually a dynamic process. And that's how they were able to identify the DNA demethylation pathway, where they started to synthesize the different intermediate molecules to get to that demethylated state of DNA. And in one of those intermediate steps, we have hydroxymethylation, which comes after the oxidation of DNA methylation. And as the research was evolving, it was pretty evident that this itself was a stable epigenetic mark that had a role in transcriptional regulation that was unique from DNA methylation. It's environmentally sensitive in that it's prone to changes to the environment, and it's very specific to brain tissue. And this is what really drew me to study hydroxymethylation. It's very highly enriched in neuronal cells. It's implicated in synaptic plasticity, involved in cancer, Alzheimer's disease, Huntington disease, but very, very few work done in the psychiatry field, and no work at all in trauma or PTSD. So for this, and in collaborations with the VA Brain Bank, we were able to study human postmortem brain specimens from the orbitofrontal cortex. And this included 25 PTSD cases and 13 controls. And I'm here showing the ancestry. The sex were all male, and the different covariates that we included in the model: postmortem interval, age of death, opioid use disorder, because these samples were certain as well for opioid use disorder and smoking. I have to say that this work has been done by our talented postdoc, Dr. Diana Núñez Rios, who took the lead on the analyses. So to study DNA methylation in parallel with hydroxymethylation, we did reduce representation oxidative bisulfite sequencing in neuronal isolates. And we did the differential analysis using the methyl kit R package, using a linear regression model, pretty similar to a standard EWAS. I used the FDR set at 0.05 as significant after multiple testing correction. Here I'm just showing how the coverage looks like in our sequencing assessment. We have an average of 10x coverage, which included 3.5 million CPG sites, which is more than three times the CPGs included from a commercially available latest version of the Illumina methylation array. Most of these, 44% of these were located promoter regions, sort of capturing those that are functional at the gene regulation level. And here I'm showing a circles plot on the left, where we show the DNA methylation and the DNA hydroxymethylation marks. Those in red are showing hyper direction and hypo on blue. And we found that 539 differential CPGs or CPGs for DNA methylation were identified, and 816 for DNA hydroxymethylation. So pretty substantial higher number of DNA hydroxymethylation over DNA methylation. And these were more mostly found in promoter regions, which indicates a potential role in gene regulatory processes. So from these genes that were identified, many of them have been pretty well known in the literature. We have, for example, DRD4, we have CRHR1, which was one of the latest hits for GWAS of PTSD in the literature. And pretty much we're showing here genes that have been very well known in the PTSD field. And again, we're seeing a lot more on hydroxymethylation than in methylation, at least in neuronal cells in the OSC. We also look at DNA methylation and hydroxymethylation in non-CPG sites. Even though methylation occurs predominantly in CPG sites, we also know that it can happen in non-CPG sites. And it's very interesting because DNA methylation in non-CPG sites is known to be highly enriched in neurons as well. So we thought we were well suited to dig into potential role of DNA methylation in non-CPG sites as well in the context of trauma. And for that, we identified 206 differential methylated sites and 159 hydroxymethylated sites in non-CPG sites. And pretty much they were all enriched for developmental processes for both CPG sites, for CHG and CHH non-CPG sites. And we also look at gene networks, combining these two data sets, and we found neurogenesis, regulation of response to stress and immune system processes as the pathways where our genes are involved in. We also show basically across the board so far that DNA methylation seems to play a bigger role than DNA hydroxymethylation, that DNA methylation in the brain. And I'm here showing the enrichment for diseases using Metascape for hydroxymethylation and methylation. And we do see a lot more enrichment, a stronger enrichment, and for more disorders. And one of these disorders include mental disorders. And from the genes here, we're mapping here in this diagram the different genes identified from our data from CPG sites and non-CPG sites. And again, just to highlight some of the genes that were known in the PTSD literature, like DRHR1 gene and GFR1 and ESR1 gene, DRD4, MPY. So genes that are known to be involved in the stress regulation system. So here we integrated data from our methylation in CPG sites, hydroxymethylation in CPG sites, and for both in non-CPG sites, but also with transcriptomic data where we actually have some sample overlap as well. So which should be good to indicate potential effects of the methylation and hydroxymethylation patterns on gene expression. And by integrating all the omics, we found that across the board, we have an enrichment of developmental processes. And by doing a protein-protein interaction network analyses of all the genes annotated from all the omics, we found, we identified different pathways, multiple immune-related pathways, and GPCR signaling as well. Oops, sorry. And we asked the question on whether can epigenetic biomarkers inform treatment for PTSD. And for that, we did an in silico drug repurposing analysis. This was done by Dr. Jaime Martinez Magaña, another postdoc from our group. And we identify drug treatments actually that has been already tested in clinical trials, such as antialarming targeting CRHR1, but also giving insight on additional potential drugs targeting CRHR1, but also DRD4, PDA4, which are genes that are also well established in the PTSD literature. So conclusions for part 2 is that DNA methylation play an important role in PTSD based on findings from the OFC neurons. DNA hydroxymethylation may play an even more important role than methylation targeting novel genes and those already well known in the PTSD literature. These were predominantly located in promoter regions, suggesting an effect on gene regulation. Convergent analysis with transcriptomic data showed a role for developmental processes. By integrating the different omics, we showed enrichment for and interactions with GPCR signaling, immune system, transcriptional processes. And our drug repurposing analyses identify promising drug targets from the treatment of PTSD. So basically our take-home messages, epigenetic modifications are associated with trauma exposure and PTSD. This effect seems to be system-wide based on our findings from periphery and the brain, and across the lifespan based on our data from children and adults. DNA methylation played an important role targeting novel genes, and those already well known in the trauma and PTSD literature. Hydroxymethylation, interestingly, may play an even more important role than methylation, at least in the brain and in neurons. These epigenetic alterations have an impact on gene regulation, and its effects may be long-lasting. And integrative multi-omic analysis identified gene networks such as GPCR signaling and immune system. And given its dynamic nature, these changes may be also reversible, opening new avenues for treatment and interventions. And here we suggest potential drug treatments or targets for PTSD. And I'm going to show 1 minute this slide. I'm also the co-founder and co-leader of the Latin American Genomics Consortium. This is very important given the lack of a diversity in genomic studies, which has been shown to potentially exacerbate health disparities in already underserved populations. So if you have any questions on this as well, or are interested in joining our efforts, please feel free to reach out to us. And I want to thank our lab, Dr. Sheila Nagamatsu, Diana Núñez Rios, Jose Jaime Martinez Magaña, Diego Andrade Brito, our collaborators from Mount Sinai and Yale and VA Connecticut, and Traumatic Stress Brain Research Group, PGCSUT, and the Human Genetics Division. So here's my contact info, and just want to also bring into awareness that Hurricane Fiona hit Puerto Rico actually 2 days before our 5th anniversary of Hurricane Maria. So if you're interested in helping Puerto Rico, that's already suffered so much trauma, including a series of nonprofit local organizations, and I can assure you that the money will get to where it's needed the most. Thank you. Again, any questions?
Yeah, so just to kick off, we have a survey. So if you can please fill out the survey, it would be greatly appreciated. So the first question was from Stephen Fleming. Is there a mechanism for methylation like this to occur as an effect of trauma, or is it thought that the observed methylation patterns are inherited?
Right. So right now we're doing methods such as LD score regression to sort of give insights on whether these changes could be inherited based on, whether these map to nearby SNPs that have been found in the GWAS literature, basically. With the idea of disentangling whether these are causes or consequences of trauma exposure, and whether these effects could be passed on to generations. So hopefully we can provide some of that data soon. But yeah, that's a really good question. And also I think we're in its infancy, and whether to really check whether these changes can be passed on the generation. I don't think we're there yet to support that, but it's a very interesting aspect of studying epigenetics.
So the next one is from Cherish Taylor. Do you see any differences associated with when the trauma occurred, childhood versus adult trauma, or when trauma occurs during both periods?
Right. So we're actually conducting now a meta-analysis of different children cohorts and different adult cohorts in saliva, just to be able to look at in well-powered, better powered data sets reached by meta-analysis, to then see how there is some convergence or not of the findings from children and adult cohorts. I mean, ideally, you would like to follow up the children cohorts up to adulthood, but I don't think we're there yet. And it's obviously very expensive to do that. But I think at least doing this could provide, hints on whether we do see some convergence of genes, and that could be potentially, giving us clues on whether the changes that could be happening early in development could be long lasting and, persistent until adulthood.
So next question is there, it's from Sean D'Souza. Is there a relationship for or influence of childhood trauma on PTSD or other psychiatric and mood disorders? Do you see differences in male and females in this cohort?
Right, so there is influence of childhood trauma on the development of PTSD. I'm not sure about sex differences. I don't think that has been studied deeply enough to have an answer, but that's certainly very interesting to look at. So far, the sex differences has been studied in the context of PTSD and genetics, has been studied in GWAS field, like doing some heritability analyses, not yet stratified GWAS analyses, but at least the heritability findings show that there are significant and substantial differences in heritability for PTSD, showing a higher risk, higher heritability for women over men. So that's certainly interesting. I think the epigenetics should cut on that area and explore potential sex differences at the epigenetic level as well.
So there's an anonymous question. Is there research that measures methylation changes following traditional talk therapy? And they say, I hope your family in Kamui is safe and has found immediate support if they needed it.
Yeah, thank you. Kamui is in the website. Yeah, thank you so much. Yeah, they're safe, which is important, and they're all together, so it's good. Yeah, so I'm not aware of any research using talk therapy and looking at epigenetics after talk therapy, but that's certainly very interesting, especially if after that talk therapy you see changes in behavior and improvement of any treatment or improvement of symptoms. Yeah, thank you.
Do you have any insights into like, are there, oh, sorry, there's one question that Danilo McCallie, thank you for such a great presentation and super interesting findings. I think I missed which was the sequencing library method used for the 5MC and 5HMC analyses. Would you mind quickly mentioning that again?
Sure, we used the reduced representation oxidative bisulfite sequencing. So this method should be targeting for CpG sites, although we also see coverage for non-CpG sites, and we had a coverage of around 3.5 million CpGs. So yeah, we do have a lot more coverage than the commercially available array, which is 450,000 probes, CpG probes. But we also analyzing new data using whole genome bisulfite sequencing. But unfortunately, this data was not able to do that oxidation step before the sequencing for us to be able to distinguish between the two modifications. But certainly, yeah, bigger data set as well. So hopefully we'll have some more data soon.
Okay, so we're at the top of the hour, and I'd just like to thank the speaker, Janitza, again. Thank you so much for coming.
Oh, thank you, Ricardo, for inviting me. My pleasure. And if you have any questions, feel free to email me. Thank you so much.